Question 6. The antibiotic bacteracin is produced by the bacterial species B. subtilis. The graph below shows a growth curve of a population of this culture to produce the antibiotic. We've got phase A, phase B, phase C, phase D, which you should recognise as a standard growth curve. Name phase A and explain why cells do not divide during this phase. Okay, phase A is the lag phase. And the reason that they are not actually increasing in that time is because they are basically getting sorted for the environment they're in. Okay, so what you've got here is that you have enzyme induction. Enzyme induction to the conditions. And that's pretty much what you're looking for. Be really, really careful because obviously this is the lag. This can also be called, this is the log or the exponential, so make sure that you can tell the difference between an A and an O. This is your stationary, and this is your death. Okay. Name the phase in which the bacteria produce the secondary. That's in phase C, but name it obviously, so stationary. Explain why the secondary metabolite gives an ecological advantage to them. Okay, so this is an antibiotic, so this is killing other bacteria, and basically that's what it's doing, is preventing competition from other bacteria. Because if it can clear out other bacteria from round about it, it has access to everything else. Okay, this growth curve shows viable cell numbers of them. Give evidence from the graph to justify this statement. Okay, so if this is a viable cell count, that is the number of functioning living cells. If you didn't have a viable cell count, basically you couldn't see this phase. Okay, so we've got a reduction in cells. So phase 4, or phase D shows reducing cell number. If you had not done a viable cell count, but you just done a total cell count, then you would have seen all of the dead cells as well, and so that would have still been increasing in number. Okay, that's the question.